Yes, good day, class. Thank you so much for the time that we spent this year together. It has been so amazing and wonderful. Uh, please check. I'm sure that you are to celebrate this year. This year have never been the same. Good predictions, good time together, and thank you so much for subscribing to Metric Hub. And I'm sure that you will never regret, but you will live to celebrate. Referring to our papers that we have written this year, Metric Hub had managed to save millions of lives there. I can tell you with confidence that for anybody who has been following Metric Hub, life sciences has never been the same. It was quite easy. You wrote the exam with a quite bright smile on your face. Just to refer you to the last presentation that I posted on paper 2 predictions, I'm sure that you saw that for those who also managed to look through and follow the presentation, paper 2 presentation, the paper came as it was like. The whole presentation managed to take into account the entire paper that we were writing yesterday. And I'm so happy for that. Experience is the best teacher. I'm so happy for you. I wish you all the best. And let's celebrate this festive season, knowing very well that we have done it. I'm just coming to you right now to confirm the marks, to confirm the performance. I'm going to present section A, section B are in different pieces because of the length of the presentations. So let's just quickly go through these presentations and you'll see. And once more, please don't forget to recommend to friends and family that Metric Hub is the channel of choice. It's all about education. We welcome you home. We are a family, and all things are so good and done for the best of everyone in the class. Okay, without wasting any time again, let me just go through the question paper that we have written yesterday, just to quickly confirm and check things better to all of us. The instructions, I'm not gonna follow them. I'm sure that this I've exhausted and I've dwelled extremely hard on that, and I'm sure that you are quite sure that the instructions, you were following them. You didn't make any mistakes, and keep it up for that. Let's just quickly mark the paper now. Question number 1, which of the following organelles is the site for protein synthesis? Proteins synthesis, which I told you last time, that is also called what? Translation. Protein synthesis, others might have think that or might also remember it as what is translation. We said that the ribosomes, it is the manufacturing site for proteins. It is the industrial site for proteins, which reads the order of codons on mRNA step by step, in a specific order, step by step. So please understand that it is the ribosome where proteins are manufactured. That is where proteins are manufactured. And in humans, the chromosomes determine gender. That is true. I also presented to you on the last presentation where I was giving scope, giving you gender or sex determination, which I was giving you the calculation of how to calculate probabilities of having a boy child or a girl child, which I also showed you that it can be done through a genetic flow diagram or it can be done through a Punnett square. So please understand that in terms of chromosomes, we know that males are XY, which is also fine, and then female is XX. So we can understand and accept that XY, XX is the chromosomes that represent our gender, which is C. It is XY and XX, which you can agree with me that it is C. An organism that is genotype homozygous T. I also told you again that this is called homozygous dominant. It's called homozygous dominant, where I was telling you that it is equals to the same as heterozygous T. Both of these will refer to mean to say they can mean tau. I said this can mean tau, and I also told you that this is the dominant allele, and then this is the recessive. This is called the recessive, which can only express if it is in their own combination. And then which I was also telling you that this is called complete dominance. Complete dominance, where we have got a heterozygous combination. So please understand, and I'm making sure that for those who are doing grade 11, coming to grade 12 next year, you are not lost. Keep following metric up and you get your things right. So this is called homozygous dominant expression. It's called homozygous dominant. This is called homozygous dominant because homo means same. This is homozygous dominant, which you can see that we have got. I said we don't talk about these as capital letters or small letters. We use dominant and recessive alleles. We don't use, that's the scientific expression of this. Down syndrome is a genetic disorder. It's a genetic disorder where an individual has an extra copy of chromosome 21, which we say is a trisomy condition. Which of the following may lead to Down syndrome? It is failure of the chromosome pairs to separate during meiosis. When chromosome pairs do not separate during meiosis, they result in what? In failure of chromosome pairs to separate during meiosis. That is to say that our answer is what? Is B. And obviously here it is what? It is C. So I also told you, I was also tipping my learners before exam that you can't go to write paper 2 without understanding the non-disjunction of chromosomes, which I was saying it is called also non-separation, which is a genetic disorder normal caused by resulting in trisomy 21, which of course causes Down syndrome. So please understand and get this right for those who are doing grade 11, coming to grade 12 next year. A gradual change in the characteristics of species over time is known as what? It is known as speciation. Speciation, I'm sure you remember the forms of the types of speciation, which we said that we have got what? Reproductive isolation, and also we have got geographic speciation, which can be caused by a physical barrier and stuff. I explained that also on our channel. Go and follow on that, and you will see that that is what you mean by gradual change in in species characteristics, which we said that there is no gene flow, which at the same time is caused because of the organisms are undergoing through isolation from each other, and then they cannot be able to interbreed. So over time, speciation will result where changes of characteristics will take place. Which of the following is a reproductive isolation mechanism? adaptation to the same pollinators. It is not isolation. Specific species courtship behavior. Organisms, they don't randomly mate, but they've got courtship behavior that matches or that is that works for a specific species of organisms. So each type of organisms has got their own courtship behavior, which attract organisms of the same species. So it is specific or species specific courtship behavior. That is the reproductive isolation mechanism. Because if you look at C, production of fertile offsprings, it is not when organisms are able to interbreed. It means they are what they are of the same species. Breeding at the same time of the year means they are not isolated. It means they are compatible, and hence there is no isolation mechanism that is in play there. Analysis of mitochondrial DNA is an example of this line of evidence. When you talk about DNA in this regard, you always have to understand that it refers to genetic evidence. Genetic evidence, it is the one that explains mitochondrial DNA. Remember, when we're talking about DNA, you cannot explain DNA and out of any other context other than what? Genetics. So it is genetic evidence, which we say that mitochondrial DNA is passed from generation to generation by what? By female organisms, because of this specific process of fertilization, where we said that the male cannot pass it because mitochondrial DNA is contained in the middle piece of the sperm cell. So yeah, that is it. Which option in the table below shows the correct comparison between mitosis and meiosis? I also mentioned that you need to know the differences between mitosis and meiosis, where I was specifying that mitosis, there is always good understanding that is expected from a learner with regards to these processes, as in terms of saying meiosis is specifically for gametes formation. Gametes formation, one thing that you should not forget, and this one we said it is specifically for cell elongation. We can say cell growth, and we can talk about obviously replacement of what, which we can as well talk about replacement of cells, cell replacement. So when you look into that, we are saying produces 4 daughter cells, produces 2 daughter cells, and then we are saying mitosis, it does not produce 4 daughter cells, it results in 2 daughter cells. That is one thing that you should know. There is no 4 daughter cells in mitosis. And then produces genetically different cells. They are not genetically different. Remember, we are saying that there is replacement of cells. And then here they are not identical. Also, they are different. So, which we talked about variation in regards to meiosis. I talked about that before again. And then chromosome number, it remains the same, which is right. The chromosome number is halved. That is also the same. I mean, that is correct. So we are saying our answer is what? Is C. We don't have 2 divisions. We have got we don't have 4 divisions here. So what we are saying is our answer is what? Our answer is T. I mean, is C. And then which of the following scientists discovered the fossil towing child? It is Raymond Dart. Raymond Darty is the one who discovered the towing child. I was saying again, you need to know this various scientists who took part or who participated in specific events and discoveries in history and evolution. So in terms of the fossil towing child, it was discovered by Raymond Dart. Which of the following is produced at the end of translation? Translation, it ends up in amino acids. Amino acids are the ones that are produced there, which combine together to form proteins. So in translation, we are saying amino acids are produced, which forms a peptide bond. I'm sure, remember when I was showing you a structure like this, we are saying this is called a peptide bond. The peptide bond, which I was saying that we are talking about a long chain of amino acids joined together here. So amino acids are produced, which I mean that these are produced at the end of what? Of translation. So 20 marks, you can get them, or you were able to get them clean and smart. Biological terms, a nucleic acid that carries hereditary information, it is DNA. DNA carries the hereditary information. I'm sure you remember about that, why, or the function of DNA. The chromosome condition of a cell that is a single set, uh, we call that a haploid. It is a haploid when it has a single set. Remember we said haploid, diploid. Haploid is when it has got a single set, and then the structure that holds the two chromatids of a chromosome. I'm sure you remember that is the centromere. That chromatids are joined together by a centromere. That is correct. And the type of RNA that carries specific amino acid to site of protein synthesis, it is what? It is tRNA. tRNA brings the specific amino acids to the site of protein synthesis. That is correct. The analysis of DNA samples to identify individuals or relationships between between individuals. I'm sure I remember the DNA profiling is called DNA profiling, and a representation of the number and structure of all the chromosomes that occur in the nucleus of a somatic cell. I also have also explained that karyotype, it is the arrangement, structure, and number of chromosomes in the nucleus of a somatic cell is called the karyotype, which we talked about. You might also have seen it as being presented to identify whether it's a male or a female, or else you were also gonna be able to be asked to say normal or abnormal person. So the karyotype is one of the key areas that also grade 12 you must not miss when you go to paper 2 exam. A group of similar organisms that are able to interbreed is called a species. When we're talking about species, these are organisms that are able to interbreed together. That's called producing fertile offsprings, producing individuals that are able to breed also. We call that a species. The phase of meiosis, where paired chromosomes are arranged at the equator, is metaphase. Metaphase 1. And the biotechnological process that produces genetically identical organisms, that is called cloning. That is cloning. So you can see in terms of the biological terms, I've been telling my class and my followers on this channel that this is one other area which a learner must get all the marks. You must not miss anything. Basically, I'm saying section A, it is 50 marks. Of automatically, the time you get 50 marks, we are not gonna have anything else but a pass. So section A ensure that you don't get anything less than 48 if you don't get all the marks. Okay. A source of variation in population, it is caused by random fertilization, also by random mating. Organisms, they do random mating also where we are talking about odd organisms and random fertilization. There is no order in terms of fertilization. So we are saying they all bring, so it is both A and B. Both A and B are correct. They are correct there. So we are saying the discovery of DNA was done by Franklin and Wilkins. It was these historians or scientists who discovered, who have got theories of evolution, Lamarck and Darwin. And then number three occurs during meiosis. These are the processes that takes place during both meiosis and what? And meiosis and mitosis. So during meiosis, we are saying A and B, so it is both. It's both A and B also in question number what? In question, in this question. So I'm sure that you know that what is it, the processes that we are talking about, the karyokinesis, which is a division of a cell nucleus and usually followed by cytokinesis. You remember that it takes place in meiosis also. And then the cytokinesis is actually a physical process of cell division which divides the cytoplasm of a parent cell in the daughter cells, and which concurrently occurs in what? In mitosis and meiosis in animal cells. In animal cells, that's how it occurs. So let's just go quickly to the next question. This is a DNA structure. We are given a DNA structure here, and then DNA molecule, which is undergoing, obviously, I told my learners that the moment you see the DNA structure that is shown here, and when it is unzipping a part of it, and then we see that this process is representing DNA replication. Weak hydrogen bonds are broken there, and we can see that this is a specific process, which is DNA replication, process by which DNA is copying itself, is producing an identical copy of itself. So identify the process shown in the diagram. I'm sure that I said that that is DNA replication. And then chemical bond labeled 1. Those number 1 is weak hydrogen bonds. That is weak hydrogen bond. Weak hydrogen bond is number 1. We said that they are there to be weak, so therefore they can facilitate the process of replication. And then next question, collective name for the path labeled 2, 3, and 4. 2, 3, and 4. 2, 3, and 4. I'm sure that this I even showed it a day before the exam, that you should know that the building blocks for DNA is what? Is nucleotides. Nucleotides. And then the full name for the nitrogen base as nitrogenous base 5. Nitrogenous base 5. You can see that this base, it is actually complementary to what? To A. So A is what? A pairs with what? With T, which I'm sure that you know that is what? Thymine. That is thymine. I talked about the complementary pairing of bases a night before this paper, a day before this paper. I showed you that what do we mean by complementary pairing? During which phase of the cell cycle does this process takes place? We say it takes place during the S phase. Number 1.43, S phase, which is called the synthesis. It takes place during the synthesis phase. It takes place during the synthesis phase. Where in the cell does this take place? It takes place in the what? In the nucleus. We are saying for all living organisms that have got the nucleus, it takes place in the what? It takes place in the nucleus. It takes place in the nucleus. And I was saying that this question is something that you must not forget. So it is one of the areas that you should know that DNA is contained for the DNA that we are to worry and explain more about. It comes from we are referring to the DNA in the nucleus. And then I was saying that for eukaryotic cells, it is in the nucleus, and then also it is in the nucleoid region of the prokaryotic cells. Those ones which have no nucleus, it is called in the nucleoid region. The nucleoid region, that is for the what we can also say or or uh or the it's in prokaryotic cells, it is found in the or it occurs uh, in the nucleoid region, where those ones which does not contain nucleus. So please take note of this, and you will never go wrong. For those who are in grade 11 following up on metric hub, keep connected. Let's just complete the question. Section A, I'm saying in a certain species of butterfly, one gene controls the wing spot color and another controls eye color. The wing spot color can be red spots or gray spots, while eye color can be black or brown. So we have got two characteristics that are, we are having two characteristics that are in question here, and then name the type of cross represented. Remember, we said when it is more than one characteristic, or when you have this is called a dihybrid cross. We have got the gene that is controlling the eye, the color of the what. We have got the one gene that controls wing spot color, and then another one which controls the eye color. So we are saying there are two genes that are being explained or talked about. It's called a dihybrid cross. Give the dominant characteristic of each. It is already given here. The dominant is for each wing spot. I'm saying wing spot color. Wing spot color, it is what? It is red spots. It is close to red spots. And also for eye color is black. Each characteristic. So I'm identifying each characteristic here. Eye color is is black. So the dominant characteristics are these ones. So we are saying it's red, spotty, and black, which are the colors or the dominant characteristics that are asked in the question. It is black and red spots. Okay. We are given the table that shows phenotypes, that shows the phenotypes or the possible genotypes of the offsprings, and then the phenotypes are given all the possible genotypes. So having that question first is saying give all the possible genotypes at X, the possible genotypes at X. We are saying red spots and brown eyes, red spots, and brown eyes. So we have got red can be like this, red spots and what, and brown eyes. Brown eyes is represented by eye color is represented by E, so it can be homozygous recessive red, and then brown is that one, or we can have also this and that. They are all correct answers with regards to the X. It can be any of these. It can be this because this means red and brown, this means red and brown. So I mean that is what we mean by this expression of the genotypes that were given there. And I told you that the genetic makeup, it means it is the genes, that is something that is the information that is contained in the genes. So you don't give it as in terms of the physical appearance, but it must be expressed in letters or in unknowns. And then the phenotype at Y, this is red and black. This is red spots, red wings or red wing spots, and black. So you can see we are saying it is at Y we have got this dominant E, R is dominant, E is dominant. So we are saying red spot wings and black eyes. That is the phenotype there. And then the last one, the genotype of the gametes produced by butterfly with gray, genotype of the gametes produced by a butterfly with gray spots and brown eyes. Butterfly that is gray spots. We are having gray spots and brown eyes. That is this is the gamete producing cell. So this gamete producing cell, it undergoes meiosis. It undergoes meiosis. When meiosis occurs, it produces or it gives us the gamete that can be formed here. So we can have a gamete which is going to have, we are saying half of this is going to be what? Is going to be RA. Is 2R and 2E. Therefore, the answer should be what? The genotype of the gamete produced is going to be, this is the gamete RA. RA is the gamete class that is produced in this case. RA is the gamete that is produced. So I'm saying, I just wanted to give you in two pieces. Section B is coming with question 2 and 3. Section A, 50 marks, you get them right, and I'm sure that you got them very correct. Thank you so much for your time, and thank you so much for following Metric Up. And I trust that you are going to recommend your brothers and sisters. How have you made it? That through Metric Hub, it gave you all the support. Everything was easy and everything was well presentable and was done. Thank you for your time. All the best again in your next academic journey. For those who are in grade 11, please, you are welcome home. Metric Hub is the home for all of us. Let's be together, and together we can do this. Thank you, and you have a good festive season.